Mendy is going to do the introduction. I just want to welcome everybody as Director of GDPE to this final invited seminar of the semester. We're very, very pleased to have Dr. Winfree here with us, and I will let Mendy take it away. Thank you for all participating.
Yes, so it's my great pleasure to introduce to you today's speaker, Dr. Rachael Winfree. I became aware of Rachel's work a long time ago, and I've really enjoyed reading her work. But in particular, what made me say, like soul sister, is that she also has a great interest in dominant species. So shout out to that, Rachael. Yay.
All right.
But so let's just see where Rachael came from. She got her BA in government, which that was cool to see that at Dartmouth. But luckily she came over to the green side and did a PhD in ecology and evolutionary biology at Princeton University, graduated in 2001, and then went on to postdoc at Princeton and then at Berkeley, and then joined the faculty at Rutgers after that, and has been there since. Rachael is a highly accomplished scientist. Her goal is to understand the causes and consequences of biodiversity, and she does that in a number of approaches. But what's really cool about her research is she tries to do this in real world settings. So shout out to that too. And she does this at scales that are often not encompassed in a lot of biodiversity studies. So she's been well funded to support her research, and she's published, I think I could figure out, it was at least over 70 papers, if not more. I might be underestimating that. And her most recent, H index from Google Scholar is 44. So people like her research too, and they cite it, which is always a nice thing. And she's published in really high impact journals such as Science, the Proceedings of the National Academy of Sciences, Ecology Letters, et cetera, et cetera. She's been on the list for highly cited researchers in the field of environment and ecology. She's a fellow of the Ecological Society of America, and she serves on the board of directors of the Xerces Society, which is one of the largest, or the largest, pollinator conservation efforts out there, nonprofit efforts out there. I'm really, really excited to hear your talk today. And of course, I don't know where I put the title, but I know it's about pollinators and ecosystem services.
Good enough. Good enough.
You're going to show it anyway, probably.
Yeah.
But take it away, Rachael. We're so pleased to have you join us. We just wish you could join us in person, but maybe we can get you out here another time so that we can meet with you in person and have that fun, the fun of interacting with people face to face.
Absolutely. No, thank you, Mendy, for that really, really nice words. So thank you everyone so much for inviting me to give the Distinguished Ecology lecture. So I've always really admired the Colorado State grad program, actually, as kind of from afar, just as a place that really excels at both the quantitative theoretical aspects of our field and also equally so at the kind of real world or biological aspects. And actually the first person in my lab, other than me, so the second person in my lab, I hired right out of your grad program. Dan Caraveau came here in 2008, I think. He did his postdoc with me, and he's now a tenure track professor at University of Minnesota. So yeah, I wish I could be there to meet everyone in person, maybe next time. I've actually spent a lot of time in Colorado because my grandparents live there. So speaking as someone who lives in New Jersey now, you all are really lucky to wake up to Colorado air and Colorado sky. So beautiful in Fort Collins. Okay, so let me share my screen. And get started. So that works. Someone give me a thumbs up. Cool. Okay. All right. So the title of my talk is What Can Pollinators Tell Us About Biodiversity and Ecosystem Services in Real-World Landscapes? So really my talk today is about biodiversity and its relationship to ecosystem functions and services, which probably all of you know, but just in case there's some people who aren't ecologists, that's things like primary production, nutrient cycling, carbon storage, pollination. So in other words, it's ecological processes that are essential to life on Earth as we know it. So the premise of my talk is that global biodiversity loss is clearly happening, but ecology as a field hasn't really answered the question of how biodiversity loss is going to impact ecosystem services. So why hasn't it? I think there are fundamentally two reasons. The first is that the focus of ecological research has been on assembled experimental communities in this field, whereas natural communities are fundamentally different in ways that matter a lot for the biodiversity ecosystem function relationship. And then the second reason is that ecological research is focused on small scales, but different mechanisms might emerge at large scales. And in addition, recent work by Mark Velland, Maria Dornelis, and others suggests that actually biodiversity loss in the real world isn't occurring at small scales, only at large scales. So most of what the field of ecology knows about the relationship between biodiversity and ecosystem function comes from what are called biodiversity ecosystem function experiments. So for example, these grassland plots here on the left. So this body of work shows that ecosystem function generally increases with the number of species in an ecological community, as shown in the plot in the upper left. So on the X, you have biodiversity, most often measured as number of species. On the Y, you have the ecosystem function in that plot. And basically, when you have more species in one of these plots, you get higher levels of function. And that's true across a pretty wide variety of functions. So my research program asks this same question, but at large scales and in real world systems. So I define large scale as tens to thousands of square kilometers. And by real world, I just mean unmanipulated, naturally assembled communities. So somewhat surprisingly, as the graph on the right shows, there really is no consensus on the answer to this question in the real world. So I think there are four things that we need to understand more about in order to know how important biodiversity is to ecosystem services in the real world. And the first few of these things are basically features of real world communities that haven't been captured well by experiments. So I'm just going to step through what those are. So first is that natural communities all have highly skewed species abundance distributions, as shown in this histogram. So this just has a number of individuals per species on the X and the number of species having that number of individuals on the Y. So basically, this means that there are a small number of common species, each of which is represented by many individuals. And then there are many, many rare species, each of which is represented by just a few individuals. So in contrast, most of what we know based on experiments comes from more even communities where most species have a similar number of individuals. And that's just so because that's the way that the experimental designs are usually set up. Okay, so second, in nature, when biodiversity is lost, species are lost in non-random order. For example, species that are most sensitive to global change might be lost first. And in contrast, the usual practice in biodiversity function experiments has been to randomize species loss order, such that all species are equally likely to be lost. And the point of this is to isolate the effect of species richness, that is biodiversity on function. But in so doing, they're also possibly limiting their scope of inference. Okay, and third, in nature, aggregate abundance, just the total number of individuals in a community, varies wildly across space. So most experimental research has controlled abundance, either directly or indirectly through, for example, standardizing the area of a small plot or standardizing the resource supply per study plot. And because of this, the role of total abundance, in other words, regardless of number of species, just total abundance, has been mostly excluded from experiments. So then there's also the issue of scale. So the research that I'm going to show you today was done at spatial scales, many orders of magnitude larger than most experiments. And when you look at large scales, you're really no longer looking within communities, but you're also looking across communities. So now we're also dealing with species turnover across space, or what ecologists call beta diversity. All right, so those are the ideas in terms of what I actually work on. So the ecosystem service I work on is pollination by wild bees, of which there are about 500 species native to my study region here on the East Coast of the US. And pollination has become one of the main model systems for ecosystem services research. And it makes sense that it is. It's a super important service. So animal-mediated pollination is required by about 85% of all plant species globally, and about 75% of the leading global food crops. So all of the data I'm going to show you from my lab today were collected in basically the same way. So I just sort of put this all together in one brief methods section. So to measure bee biodiversity and pollination function in the field, we first net collect bees that are visiting flowers of our study plant, whatever that is. And then we bring those bees back to the lab to identify them to the species level. So this all these individuals of whatever species, this is our measure of the biodiversity of the bee community that is doing the pollination on that plant at that place. And then in a second step, we measure the amount of pollen that each kind of bee deposits in a single visit to a flower. So we measure that in the field with free living bee communities, but we use experimentally pollinator excluded flowers. So we know they weren't pollinated before, and then we let the bee visit them just like it normally would. And when it flies off, we bring the flowers back to the lab and we count the number of pollen grains under a microscope. So to estimate the total pollination provided by each bee species, because that's kind of where we're trying to go with this, is function per species, we just take the product of the number of individual bees that we observe visiting flowers times the number of pollen grains deposited in one visit to a flower. So that gives us pollination provided per bee species in units of pollen grains, and that's our ecosystem service. So in the rest of this talk, I'm going to show you some papers from my lab that use these real world data to explore these 4 issues empirically. And basically what we're asking is, do we need biodiversity for ecosystem services in the real world? So the first paper I want to highlight for you used data on the biodiversity of native wild bees that were in crop fields, and we measured the pollination that they provided to 3 kinds of crops, to watermelon, cranberry, and blueberry. So in this paper, we used a version of the price equation from evolutionary biology that my co-author Jeremy Fox developed for use in biodiversity ecosystem function research. And I'm not really going to go into the details of that. I'm just going to present a simplified sort of summary of what the price equation actually does, because it's super, super handy for this kind of research. So we start with just the difference in blueberry pollination between these 2 farms. So these are 2 of our blueberry farms. We measure the total pollination at each farm. We start with that difference in pollination. And then we partition that difference in the ecosystem service into components that are attributable to differences in the bee communities that were pollinating those 2 farms. And this partition is into two quantities, basically. One is differences between the bee communities in species richness or composition. So that is to say, differences between the farms in the number and identity of bee species that were found at the farm. And the second component is differences between farms just in the abundance of bee species that were found at both farms. So this latter quantity has nothing to do with species. Someone would say it has nothing to do with biodiversity. It's just whether you have more or fewer individual bees of common species. So to cut to the chase here, what we found in this paper was that the effect size of that species term in terms of explaining difference in pollination received among farms was only 15% as big as the effect size of abundance. So in other words, the main reason that one farm receives more pollination than another farm is just that it has more individuals of a common bee species. It's not that it has greater biodiversity, that is more bee species. It's not even that it has different bee species. So why? What is the mechanism? So rather than report results from the paper, I want to show you some pictures I put together to illustrate the mechanism, because I think it could be quite general, not only for pollination, but other kinds of ecosystem services, given the ubiquity and nature of skewed species abundance distributions. So here I'm showing you for these 3 crop plants we studied what I call species function distributions. So these are analogous to species abundance distributions. And for these data for crop pollination, they map very closely onto the species abundance distributions. The correlations are like 0.8 to 0.9. But what these histograms are showing is a total amount of pollination provided by a bee species on the x-axis, and then the number of bee species providing that amount of pollination on the y. And these data are summed over each entire study system. In other words, I'm giving you one frequency histogram for each plant. And that's why there's such large numbers of pollen grains. And also you notice really large numbers of bee species, a very biodiverse system that I work in. So in general, a few species provide much of the pollination, and these are highly abundant species. And in fact, that species, the outlier for the watermelon and cranberry is the same species. It's Bombus impatiens, which is kind of like a weedy, what bee people would call a trash bee. It's definitely not a bee that is in need of conservation. Okay, but then in contrast, most species provide little pollination. So these species here, the great majority, are rare species. Sometimes they're just represented by a few individuals in the whole study system. So what this means for the importance of biodiversity to pollination is similar for these 2 systems, the watermelon and the cranberry. I realize that's obvious to me because I'm a pollination ecologist. I should have put the fruits on here, but it's different for the 3rd system, the blueberry. So I'm just going to talk about these 2 together first. So here, this functionally dominant species is present essentially at all sites. So it doesn't contribute to changes in the number of species across sites. It doesn't affect richness because it's always there, right? But it does vary in abundance up to 100-fold across sites. And so the abundance fluctuations of this common species is really what drives the total pollination differences between farms and pollination. And in contrast, these rare species each contribute little to pollination, but being rare, they're not present at most sites, right? So the change in number of species between sites is really driven by these rare species as they're kind of swapping in and out across sites. So what this means is that ecosystem services are decoupled from biodiversity by the shape of this species function distribution. So I think this is actually a really important, possibly even profound thing. And I think it could have wide generality across many kinds of ecosystem services, though not all. And again, it's because it's such a universal feature of ecological communities that we have a few common species and then many rare species. All right, so I said the 3rd study system was a little different. So here it is, this is blueberry. So in this system, there were more dominant species, not just that one outlier. There were sort of 9 species that were reasonably abundant here and important to pollination. And it was actually the species richness, but just of these dominant species, that was the strongest predictor of how much pollination a farm got. In other words, how many species out of these 9 that a farm has predicts how much pollination it gets. So technically species richness is important here, but still 9 important species out of about 40 overall isn't really what ecologists mean by biodiversity. Okay, so to sum up this paper then, when we looked at what features of pollinator communities explained why some farms got more pollination than others, we found that the abundance of a few common species explained most of the variation in pollination between farms. And the number of species, which is what ecologists usually mean by biodiversity, wasn't particularly important. And that's just because most species were rare and didn't actually contribute much to pollination. All right, so the next paper that I'd like to share with you focuses on the issue of spatial scale and how increasing the scale of the study changes the need for biodiversity. So the question we set out to answer here was, does biodiversity become more important to ecosystem services at larger scales? And by asking this question, we're actually changing the question. We're actually changing the axes with respect to what biodiversity function experiments have traditionally asked. And I just want to be explicit about that. So this figure shows the usual question, which is as you increase the number of species you have on the X within a study plot, how does the level of ecosystem function change in that plot? So these kinds of figures are usually interpreted by looking at the asymptotic level of function, which is W here, and reading the graph down to the x-axis, and then you say the number of species you need for asymptotic function is X. So this is a question about what goes on within one ecological community. And the main mechanism here that drives this shape of this curve is thought to be niche complementarity among the species in the same community, either in terms of how they use resources or how they perform the function. So basically the slope of this green line is driven by complementarity. It's steep if you have a lot of complementarity and different species contribute to function in different ways. So in contrast, when we move to the landscape scale, we're really talking about a different figure. So the x-axis is now the number of sites or the number of communities. And the y is the number of species needed to achieve some level of function, which is now implicit, it's not explicit on the graph, at all those sites. So this is an accumulation curve, basically. So we're saying to maintain function at n sites here on the x-axis, we read up and over to the y-axis and we say, okay, we need y species for that. So to make this graph, you have to define some threshold level of function W. So for me, that's the threshold level of pollination that must be met at all sites. And then the slope here, the slope of this blue line is driven by species turnover across space. Specifically, it's driven by turnover in the identity of the species providing the function. So if the same function is provided by different species in different places, you get a steep slope here. So the goal of the paper that I'm going to show you was basically to plot this figure with real data. So we used our same data on wild bee pollination of watermelon, cranberry, and blueberry crops. So I'm just showing you the map here because this is very explicitly a spatial question that we're asking. So the farms here are shown in colored points, and these farms are spread out over this roughly 1,000 square kilometer area between New York City and Philadelphia. So there's several important differences between the approach that we take here and what I showed you from the other paper a few minutes ago. So first, the accumulation curve that we plot here is of important pollinator species across all 48 of these sites. It's not a pairwise thing. And at each site in each year, we basically choose the minimum set of bee species that are needed to provide a threshold level of pollination to that site in that year. And then our accumulation curve takes a sum of species that are important at any farm in any year. And the reason I did it this way is I thought this might better fit what people actually mean when they talk about scaling up biodiversity ecosystem services research, which is a phrase that's used a lot in ecology, but it isn't always really clearly defined. So what that phrase means here in this analysis is that for an ecosystem service to be provided across a larger area, it means that it's provided adequately at each place within that larger area. So these are our results. So these are the exact same axes as the figure I showed you before. We have sites on the X and number of bee species required to pollinate all those sites on the Y. But now we're plotting it with real data for each of these crops: watermelon, cranberry, and blueberry. And the dotted line up there at the top is the total number of bee species that were observed in that entire study. And the different colors are different values of that threshold. I was called W on the earlier plot. So it's basically the threshold level that needs to be met at each site. And we basically did sensitivity analysis on that. We did a percentage of the mean observed value across farms and just showing 25, 50, and 75% of that. And the idea is here, it's a range of pollination levels that a farmer might want. Okay, so I'm going to zoom in on a figure that actually includes the data from all 3 crops. This is putting all 48 farms on the same plot here, just to make a few points. So you can see that these curves increase, meaning we need more bees to pollinate more farms. And in other words, it's not just the same bees at all the farms. And we also need more bees if we want to achieve a higher level of pollination. So the purple line is higher than the black line, is higher than the orange line. So both those things are quite expected and intuitive. We also need a lot of species. So here to reach the 75% of the mean threshold at all 3 crops, at all farms, we actually need almost 80 bee species out of 100 or so that we observed in the study. So this approach to the question gets a really different answer from what we've seen previously. We're now saying we need most of the bee species in the study system if we really want to provide pollination at all farms across this large area. So to explain what's really going on behind that result, I want to just show you a closer look at the data itself. So the first thing to realize is that there's species turnover between farms in both functionally important and less important or rare bee species. So to make that point visually here, I'm just showing you the species composition for an arbitrary 4 farms with these pies here. So the species that did most of the pollination at each farm are shown in colors, and the species that didn't actually do a whole lot are shown in gray. And you can see when you just kind of eyeball the colored pie slices that there's somewhat different important species at different farms. But there are also some species that are at all farms, like the light green one. So this figure shows basically the same pies, but they're now arranged into bars so that we can see all the site years in the study. This is just for the watermelon. So now each bar represents all the pollen that was deposited in a particular site year. And the pollen deposited by different bee species is shown in different colors. So you can see just from the color key, we really have a lot of bee species here, which is pretty cool, independent of all the other stuff I'm talking about. So the bee species depositing the most pollen within a site year are at the bottom. The big color bars are at the bottom. And the species that didn't deposit much pollen are those short color stacks near the top. So for this analysis, we considered all the bee species that are needed to reach the pollination threshold in a given site year as those that are necessary for that site year. So the pollination threshold is a horizontal black line. So basically all the species that are under the black line would be counted in that accumulation curve. So the first point to see here is the identity of the species doing most of the pollination. Those really big bars change some across farms. So the color of the big bars at the bottom changes some, but it's also that blue-green species that's at most of them, that's Bombus impatiens. And the second thing to see is that there's really big variation across farms in the total amount of pollination they get. The total height of these bars is wildly variable. And so I want to zoom in a little bit on some of the farms that don't get much pollination. So you can kind of see how that pans out. So these are farms that are at or below this threshold. So when we use this analysis method, many or all of the bee species found at these farms will be counted as, needed to meet the threshold because they're below the black line here. So that includes a lot of rare species, the really short bars at the tip that aren't really doing a lot of pollination in absolute terms. And then conversely, if we go to the other side of the figure, these farms that exceed the pollination threshold only need a single species to meet the threshold because they're getting tons of pollination and a lot from a single species. So that's sort of what lies behind that accumulation curve that is shown as a result in the paper. And this is just the full figure for all 3 study systems, so you can see what that looks like, right? Conceptually the same. Okay, so to sum up this paper then, what we found was that because there's somewhat different species pollinating the different farms, the number of bee species we need to pollinate all the farms keeps increasing as we consider increasingly larger scales. But at the same time, it's also true that many of the bee species that we count as needed in this analysis actually aren't doing much pollination. And the reason it happens that way is that we're using a threshold, and all the species required to meet that threshold are counted as biodiversity that is needed for ecosystem services. Okay, so in the time that we have left, I wanted to try to answer 3 questions. First, why did I get different results from 2 analyses using exactly the same data? Second, why does any of this matter? And third, as scientists, how should we even be asking this question? That is, what is the most relevant way to measure biodiversity and its impact on ecosystem services in the real world? So to take this one first. So what I just showed you, what I just showed you for this is that in my lab, we published 2 different results from basically the same data. So at first that stressed me out. I sort of felt like it's part of my job description as a PI to not let that happen. But then I decided it's actually good in this case because in effect, what we did is controlled the data and tested the effect of the analysis approach. And what I'm really interested in is figuring out what way of asking this question, what analysis approach really makes sense. Like what's analysis methodology corresponds to the question that people really want to ask about, scaling up biodiversity ecosystem services, right? So in short, what we found was accumulation curves over space show we need a lot of species, most of them, but pairwise comparisons between sites show that we need only a few common species. So then I asked myself, what do accumulation curves actually do differently that results in getting a really different answer for the greater importance of biodiversity? So a couple obvious points about that. One is they take the sum instead of the mean. Another is that they track the identity of species. And the third is that they use a threshold instead of a proportion. So accumulation curves are really asking how many species are important somewhere, not what species are important on average everywhere. And in doing so, they come up with a much bigger number, but they also include many species that in fact don't do much pollination. All right, so why does any of this matter? This whole question of biodiversity and ecosystem services. So I think it's important right now because the argument is being made that biodiversity itself is important precisely because it's what provides ecosystem services. So that argument, which isn't one that I personally support, but it's just a fact that that argument has really taken over the conservation world. And in particular, it's generally assumed that biodiversity is essential for ecosystem services precisely at the very large scales that I've been talking about today, because those are the scales at which policy and conservation take place. They don't take place at experimental plot scales. So just some examples of how powerful this concept is in the conservation world. So the United Nations Panel on Biodiversity, which is kind of the analog of the IPCC for climate change. So that panel is called IPBES, or the Panel on Biodiversity and Ecosystem Services. And so it basically treats these two things as the same issue. You know, biodiversity and ecosystem services are a combined issue. Second example, the Nature Conservancy, which is by far the largest conservation organization in the world, has largely adopted an ecosystem services argument for why what they do is important, you know, why biodiversity conservation is important. And this is just a photo of the former TNC CEO, Mark Tursek. He was originally a partner at Goldman Sachs and then came to run the Nature Conservancy. And his book, Nature's Fortune, basically is you know, all about making this argument that ecosystem services are the reason for biodiversity conservation. Okay, so lastly, how should we as scientists be asking this question? So what my lab's recent work shows is that pollinator biodiversity either is or isn't important to pollination, depending on how you ask the question. So I think the real question is, you know, what question do we want to ask? What do we mean by the relationship between biodiversity and ecosystem services in the real world? So, you know, what do we think is meaningful? What do we want to measure? So I don't have an answer to that question, but I do think that as ecologists, we need to be as clear as possible about what our data show, and especially about what our analyses are actually doing under the hood and what they're actually showing us. Okay, so I just want to finish up with some concluding thoughts about each of these four factors that I had said I think are key to sort of understanding these issues in the real world. So first, to kind of take these two together, the species abundance distribution and non-random species loss. So in nature, both the species abundance distribution and the species occupancy distribution, which just means like at how many sites do you find particular species. So both of these are highly skewed, and furthermore, the common species, the top species, tend to be the same species in both these distributions. In other words, if a species tends to have high local abundance, it also tends to be widespread. So if the function provided by a species is predicted by how many individuals there are that species, then this pattern leads to both a small number of species being needed at any one site, which is a low intercept on the graphs we've been looking at today, and also low turnover in the identity of these functionally important species, so that across sites, so that would be a flat slope on the graphs that we're looking at. And furthermore, the species abundance distribution can be a mechanism that breaks the association between biodiversity and function, and it's actually through non-random loss order that it does that. So specifically, rare species are more likely to be lost, but richness can change a lot when those species are lost, but function doesn't change very much. And within the field of ecology, experiments and also, importantly, the associated theory has largely missed this because the experimental design has used random community assembly. All right, so something about aggregate abundance. So I actually think that just neglect of aggregate abundance in ecology has been both a function of study design and experiments versus the real world, and also the narrow spectrum of study systems that are used in biodiversity function work. So most biodiversity function studies have been done with grassland plants or microbes, and these are both immobile communities. And also experimenters generally standardize plot area and resource availability per plot. So all of these factors end up controlling variation in aggregate abundance. But in the real world, and particularly with mobile species like animals or pollinators, resource availability varies wildly across sites, and the animals track that. And as a result, you get these radically different aggregate abundances at different sites. So it's a valid question. Is the real world abundance variation noise with respect to the biodiversity function question, or is abundance actually part of biodiversity, just one that, you know, maybe we haven't focused on enough? And I think it's actually part of biodiversity. And in fact, abundance is a part of biodiversity that's most clearly declining due to anthropogenic effects. So I would certainly think we're losing ecosystem services with declining abundance. And I think that's really important, even if it's nothing to do with species. So in a way, species is an odd measure of biodiversity decline, because you don't lose species until the very end when you lose the last individual, right? So it's not a very sensitive metric. From a conservation point of view, it doesn't really give you a result until it's too late. So I would argue that just abundance of individuals is very much a part of biodiversity and should be counted as such. And I think that even if I'm wrong and it ends up not driving ecosystem function, I also think it's really odd that all the diversity metrics that ecologists use, not only traditional Shannon diversity, Simpson diversity, but also hill numbers, they eliminate aggregate abundance because they normalize by it. So we normalize out aggregate abundance, report only relative abundance. So we've kind of prevented ourselves from detecting the effects of abundance in, you know, the whole field of biodiversity, really. Okay, and lastly, just something about large scales. So I think ecology is too complex for mechanisms to be the same at large and small scales sometimes. I mean, really, we're talking about many orders of magnitude larger when we go from field experiments to landscapes. You know, we're going from experiments which are maybe tens of square meters to thousands of square kilometers. So I think it's, you know, it's probably not too surprising that we're seeing different mechanisms emerge. Okay, that is all I have to say about my research. I'd like to just conclude by thanking funding agencies that have supported this work and acknowledge my outstanding lab group, past and present. You probably guessed that's present, but the past lab groups had a lot to do with it. And also something that I think doesn't always get said enough, which is to thank the bee taxonomists who help us identify this difficult species in our collections. So we have 99% of our individual specimens in my lab have identified as species, and that is an unbelievable amount of work. I really think without accurate species ID, biodiversity research is a little bit garbage in, garbage out. So my research program is really reliant on my collaborators. So this is John Asher, Jason Gibbs, Sam Droege, and recently Joel Gardner. So thank you. Thank you so much for inviting me, and thank you for showing up.
Thank you, Rachel. That was a great talk.
Thank you.
So I think we can open up the are we going to do it through the chat? What do you think, Ruth? What's easier?
Either way, it's great if people feel comfortable speaking up. That would be fantastic. But if you don't want to, it is also absolutely fine. Or if your, you know, kids are screaming in the background like mine just were, yeah, to write it down in the chat. Yeah. Questions in the chat.
Can you take the screen share away from me? Somehow my computer is not letting me stop sharing. Huh. All these new Zoom things we get to learn today.
I will try to do that.
I think I got it. I think I got it. Yeah, I got it.
Okay, so first in the chat, can you, Rachel, you want to look at John Mola's comment and question?
Sure. Oh, yeah. Yeah, I shouldn't call anything a trash beast, sorry. Yeah, the local and landscape features were hard to, I should read the question in case people aren't reading it. Okay, so the question is, since Bombus impatiens seem to explain the bulk of pollen delivered at many sites, what explained abundance of Bombus impatiens? were there particular local or landscape features related to the abundance of the common bee? So really good question. You know, in this study system, the factors controlling why you find a lot of bees in one place and not another are frustratingly elusive. And basically what we get here is we get a lot of bees, either species or individuals or both, at most places. And it also, we just have very high bee biodiversity. So we have very heterogeneous landscapes here. So our agriculture here is basically small family type farms. And it's set in very heterogeneous landscapes that have like little patches of woodland, little bits of development, some agriculture. And it's overall, the landscapes are really good for bees, I think. And we don't really have big gradients that seem to explain why you find bees some places and not others. And that's a kind of a foggy answer. And that's because I think it is a little bit foggy.
Great. It looks like Enping has a question for you.
Yeah, Rachel, thanks for this great talk. And I'm not a bee person, you know, so but I'm curious on one thing. So, and so even for one site, have you looked at that the variation, the abundance of variation from year to year? So that means, so yeah. The question is that, so if so maybe that in 1 year that the most abundant species or the second abundant species is one particular species, and the next year they will change because that is from the dry year to a wet year, or from the cold year to a hot year, they might be different. So that might
totally.
Yeah,
I totally agree with that. Yes, we're actually looking at it now. Postdoc in my lab is writing that paper right now. The challenge is having a long enough time series. It's easier to get replication across space in a 3-year grant than it is to get replication across years in a 3-year grant. You know, that's why we have 48 sites, but you know, we really only have 3 or 4 years on these studies. So the answer over time is the accumulation curve, at least what we're seeing so far, is not as steep as it is over space, but it definitely occurs, you know, like the accumulation curve at the same site over time. The other factor that I have wanted to look at for a long time, and we have looked at it, and we papers in review, something that bothers me about a lot of ecosystem services work in pollination is when we do these bee community scale measurements, we tend to do them on one plant, and that's just because it's an incredible amount of work. I mean, it took us like 4 years to get these data on one plant, but the reality in nature is a lot of the turnover in bee species is actually across plants, like different bees pollinate different plants. I mean, kind of obvious to anyone who's ever been outside, but is excluded from a lot of biodiversity function work actually. So we have a paper that's in review now that's looking at a natural plant community with 20, 30 plants, and the accumulation curves of bee species you need to pollinate those are much steeper than they are on one plant across space.
Thank you, Rachel.
Yeah.
It looks like there's another question in the chat for you, Rachel.
Okay. So the question is, scale is such an important factor for ecological studies, yet most are at a small scale. Do you have any advice for early career ecologists on how to conduct and manage projects that occur at large spatial scales? I don't know about advice, but I can tell you kind of what people need to do, I guess. Whether it's the right choice, I don't know. So it's basically a trade-off between having a lot of replication in space and looking at many sites across a large area, and the level of detail and the data that you can collect at any one of those sites, right? I mean, and unless you have infinite funding, that's just going to be the trade-off. So I've certainly, pollination biology in particular, this is more going into the advice mode. Pollination biology, like traditional pollination biologists, are very, very careful, precise researchers who have a culture in the field of doing things really, really well and really knowing their plant and their insects and measuring everything. And you just, so, I have had some pushback with my papers with, well, you didn't do that, measure all this stuff. I'm like, well, I also had 40 sites, you just kind of can't. So just be prepared for that. So I think that's not to say that either of those approaches is better than the other. I hugely admire a lot of the experimental, very careful work, hugely admire it, but you just can't do both. So you have to kind of have good enough, interesting enough questions for what you're doing that it can come across as interesting, even to people who aren't used to working at large scales.
Great, thanks. I'm sorry, Ian Pierce, I haven't seen your hand up, so please ask your question.
Yeah, thank you. Hi.
Hi,
So
Ian.
my question actually, that I was going to ask about variation over time, which somebody amazingly asked that already.
Yeah.
That I was actually interested in is there actually any trade-off in this system when you're managing for bee diversity versus the abundance of a really already abundant bee? So I guess thinking if I were one of those farms on the sort of left-hand side of your abundance curve, and I was like, I need to get more pollination of my blueberries or whatever. Would I actually be doing anything different if I wanted to get more of those more species or more rare species versus getting more bombus impatiens or whatever?
Yeah, that's such a good question. No, our ability as practitioners to manage bee populations at all is we do not have such ability to put it bluntly. So no, they're like basic things that are good for bees, like not using pesticides that are really toxic to bees. That applies to abundance and species, right? Having nesting habitat, same. Planting flowers that bees can feed on during the time of year that crops aren't available. Same. It's just like all generally good for bees as a thing, right? We definitely do not have the ability to manage for a particular species even. So in that sense, this whole sort of minimum set approach, which I use and which many people in this field use, is sort of nonsense, right? Because like people can't choose a minimum set anyway. Like we have no ability to manage for that.
Let's go. It's kind of a nice thing too, I guess, like working on a rare bee, like the things that people do for that might actually be good for pollination too, although like the connection is not through that rare bee.
Yeah, that's a really good point. And actually, I think the native bees benefit from people who don't want to harm honeybees, which are an exotic, invasive animal, right? But they do have some of the same needs that native bees do, like not being sprayed with insecticides, for example. So native bees definitely benefit from that. In terms of rare bees, I should say one thing though, there are a fair number of bee species. Maybe 20% of species in my system, higher percent in arid environments actually, that are specialists on particular plants. And they will definitely diverge in their habitat needs from bees that are generalists, no question.
I had a question for you, Rachel. So, thinking about kind of the next step, right, with this research is how some would argue that maybe those rare species bring about stability over time, maybe in services that are provided.
Yep.
I don't know if I necessarily agree with that, but I guess so in a related topic, it's like really trying to get an idea of this kind of pattern of our common species common everywhere.
Yeah, exactly.
Rare everywhere,
Yep.
that's such an important thing. And I would imagine it's so much harder to figure out with your mobile species rather than.
Yeah. No, but it's a really, really good question about the over time thing, because the whole idea of, the insurance hypothesis as it's applied over time is precisely that the species that's rare today may, climate conditions are changing, right? Maybe it's going to be one of today's rare species that becomes the dominant species 20 years from now. That seems very plausible. The challenge in testing that empirically is just the nature, like needing the long time series. It's,
Yeah. Thanks.
I had a question if there's time for one more.
Sure. Yeah, that'd be great, Ed.
So thanks for the talk. We read your papers as a class yesterday and had a good discussion, and we're looking forward
Thank
to
you.
tomorrow. The thing that I'm struggling with is in the large scale paper. So when you say turnover, this refers to changes in membership among sites. Okay, so that was my first challenge. I was like, turnover as temporal component. No, it means changes in membership among sites.
Turnover across space. It's analogous to across time. So, yeah.
Good. So that helps. Because we were talking about that yesterday and I kept stuck. So it's an observational study though. And then the result is that to reach this threshold of pollination at this scale, what I observe are an increasing number of species. But then the way it's presented is with some causation I just wonder about stochasticity of communities in space and founding effect. And, it's not to say if we lose those species, you would move below that threshold necessarily, because
Right. No,
that
that's
role
right.
may be replaced by somebody else. So it's
That's
just,
right. Yeah, that's right. So it applies, the sort of our scope of what we're doing applies to what is happening in the real world right now. It doesn't apply to if you took some species out, say from, a site with a lot of species, then would other species rebound, right? What people call density compensation. So that's something that one would have to check experimentally over time. And we haven't checked that and didn't intend to address it, right?
Yeah. So the comment is to achieve this level of pollination at this scale, you need this number of species.
The way that the world is right now. Yeah.
This is and it's not necessarily that we're going to move one way or another on that trajectory with loss of abundance or.
Correct.
Okay. That's good. Right. Thanks so much. Yep. That really helped.
There's another question in the chat.
Okay. Do you have any measures of the pollination effectiveness of the individual bees of different species rather than the aggregate of species? So are there particular species that are more rare, which have an outsized pollination effect of individuals, while more common species may have small pollination effect per individual, species of high conservation concern? Yeah, so we do measure the pollination deposition per individual. We can't do that for every species though, mainly because you just can't get replicates for the rare species basically. So we do it in groups of species, morphologically similar groups. So the main things that determine that per individual pollination pollen deposition is the body size and also the hairiness of the bee, like basically the big hairy bees carry and deposit more pollen. So yes, there are differences. So bumblebees are much better per capita pollinators than the smaller bees. But that doesn't, I don't know that I don't have the data to map that statement onto the axis you're interested in, which is rarity and commonness, precisely because a bee that you observe like 1 in 1,000 bees, I can't get the pollen data for it because it wasn't there the day that we were collecting the pollen data, right? So I can't actually answer that question, but in general, the situation where a rare bee species would be a really, really important per capita pollinator would probably come up not with crop plants like this, but with specialized plants, which sometimes do have a specialized pollinator. And it usually has to do with actually the way that the bee visits the plant, like how it behaves on the plant and how it contacts the anthers and the stigma. There's some plants for which your sort of average bee visitor doesn't pollinate very well. But that wouldn't be with these crop plants.
I have a quick question. Do you hear me, Rachel? Okay, so yeah, so thank you for the good presentations. And looks like your studies are more on some of the important crops, 3 species, right? And also your studies only in the eastern part of the US. So let's say some of the humid areas. So my question is like, how do you generalize your result to, let's say in the wild plants and also in some other ecosystem like arid ecosystem, where there are the chance like the wild biodiversity is very high compared to the eastern areas.
Yeah, for sure. No, you have much higher native bee biodiversity in the West than we do in the East, for sure. So I actually think the most relevant way to generalize to communities of wild plants, so you can't really get this kind of data for a whole lot of plants. I mean, just these 3 crop plants here, which are relatively easy to work with, was like 10 years of data collection. So, you know, I don't think it's realistic to like try to get the data for many kinds of plants. But what we have done, and this is a paper I mentioned that's in review, is to just look at how steeply the number of bee species you need accumulates as you go across native plant species, even in one community, because of course native plant communities have many more plant species than 3, right?
True.
And that accumulation curve is very steep. And that was the best way I could think of to kind of make the point that ecologists are, you know, all intuitively aware from our field experience, which is that at the community scale, you need a lot of species because of these networks of interactions, right?
Sure. So yeah, and also like I'm kind of interested in the habitat. Let's say, I mean, your study is more in the agricultural habitat. So,
Yeah, it was.
I mean, if you come to the West, then we don't have that. Like, let's say at least we have some natural areas and some sagebrush communities. So I'm not sure that because your pattern is going to be also will be in our west sides.
Yeah, I mean, I don't know how different this type of study would be out west. I actually think for plant pollinator relationships, a lot depends on the plant you choose, actually.
Okay, great, thank you.
Yeah.
Okay, I think we better wrap this up and let Rachel maybe eat dinner, perhaps. I don't know, but first I want to apologize. I forgot to like look at the participant list to see the hands raised. I was thinking I was in Teams, but whatever. That's what we get for switching between all these different platforms. So I apologize for those of you that had your hands up for a long period of time. But let's thank Rachel again for a great talk. Yeah, thanks so much.
Yeah, thank you so much for inviting me. I look forward to meeting people tomorrow.
That was so great. I want to go out and measure abundance of all the things now. And I just want to remind those of you who are here that tomorrow is the deadline for submissions for next year's speakers. So turn those in. Thank you so much.
All right.
That was fantastic.
Yeah, thank you so much for inviting me. Really enjoyed it. Look forward to seeing everyone tomorrow.
Yeah.
Okay.